Hello, everyone, and welcome to today's webinar, Spatial Single-Cell Characterization of Alzheimer's Disease in Mouse and Human Hippocampus. I am Matt Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by NanoString Technologies. To learn more, visit nanostring.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. Our speakers will answer any questions via the email address you provided at the time of registration. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Vikram Devgan, Business Unit Lead Spatial Genomics at NanoString, and Miranda Orr, Assistant Professor, Gerontology and Geriatric Medicine, Wake Forest University, School of Medicine. Vikram, you may now begin your presentation.
Hello, everyone. My name is Vikram Devgan. I'm a Senior Director of Product Management at NanoString and cover product portfolio of GeoMx DSP as well as CosMx SMI. I'm really delighted today to talk about our second spatial platform, CosMx Spatial Molecular Imager. CosMx Spatial Molecular Imager has been developed to provide high-plex RNA and protein quantification and visualization at a single cell resolution in a morphologically intact tissue. And let me start this presentation with a brief overview of NanoString. So NanoString is dedicated to develop spatial biology solutions suitable for your research needs. We are the only company with a comprehensive spatial ecosystem with 3 spatial solutions. NanoString offers CosMx platform for unbiased spatial single-cell discoveries, GeoMx DSP platform for unbiased whole transcriptome profiling, and Atomics as an integrated informatics portal for both GeoMx and CosMx platforms. Both CosMx and GeoMx platforms are backed by NanoString's core competence of probe design. We have more than 15 years of experience in probe design and successfully designed hundreds of predefined assays panels and thousands of custom probes across various species, which goes beyond human, mouse, and rat. Above all, NanoString is a trusted partner for you. We have more than 150 customer service and support persons all over the globe. Their whole job is to support persons, to provide you the best customer experience. No wonder we have over 350 GeoMx systems installed and over 150 peer-reviewed publications. Now, many KOLs are pre-booking CosMx platforms as we are getting ready to ship CosMx later this year. And when we looked at CosMx, one of the main differentiation for CosMx is it has the largest panel of any in situ imaging platform out there, and that's 1000-plex. With 1000-plex, you have enough real estate for cell typing, genes, and biological pathway genes covering cell state, ligand and receptors, and most informative signaling. I have used CosMx Mouse Neuroscience Panel as an example here. It includes approximately 250 genes covering 40 unique cell typing and approximately 700 genes covering 44 different biological pathways, covering different biologies all the way from neuropathology, inflammation, neurotransmission. And even after this, if we missed your favorite gene, you can add up to 50 additional genes into this CosMx Mouse Neuroscience Panel. And actually, a lot of work went into designing these panels. we have a dedicated bioinformatics and biological content development team for panel design. And if we look at the whole process of panel design, it can be split into two parts. First, we use data-driven approach for selecting cell typing specific genes. We use multiple single-cell RNA sequencing datasets with curated cell types as a starting point, and select genes based on their ability to distinguish one cell type from another. And the second focus of our panel design is enriching important biological pathways. For this, we start with curating by applications from key data sets and literature. Then we use data-driven and text mining to enrich genes that explain most variants in each pathway. At the end, you will have a panel with the most relevant gene content for covering your broad cell typing as well as biological pathways. In fact, CosMx predefined panels provide the best-in-class cell typing. Here we use CosMx Human Universal RNA panel to do cell typing with kidney tissues and compared results with Human Cell Atlas kidney data set. And as you can see over here, CosMx has identified all 33 different cell types which are identified by Human Cell Atlas single-cell RNA-seq dataset. And on top of that, CosMx also provides spatial location for all identified cell types. In addition, CosMx identified 4 different cell types and actually 4 different cell subtypes reported by Human Cell Atlas, even though these subtypes have very similar expression profile. And this is made possible due to the CosMx proprietary cell typing algorithm. Our algorithm approximately weighs the evidence for all 1,000 genes and not just depend upon few marker genes on the panel. And the spatial location of all identified 4 cell subtypes aligns very well with their assigned biology, as shown in this slide. So let's take an example of a proliferating proximal tubule subtype. It is highlighted here as a red dot. So all the dots here is a one specific cell type. And if you focus on the red dots, which are showcasing proliferating proximal tubule subtypes, this subtype is widespread in a highly inflamed kidney shown in a leftmost image. Whereas in a rightmost image, proliferating proximal tubules are aligned with injured ducts. And actually, if we take the overall view of CosMx cell typing ability and compare it with the single cell RNA-seq, CosMx cell typing actually outperforms single cell RNA-seq cell typing. And we have identified 3 different subclusters of myofibroblast, which was missed by single cell RNA-seq. And this is because in addition to the 1,000 genes which we have in our CosMx panels, CosMx algorithm can make use of spatial context and cell imaging data like cell size and immunofluorescence stains. And this overall result is very impressive. Just consider that CosMx resulted in a better cell typing than single cell RNA-seq dataset. And I personally believe that cell typing is the most fundamental or the first step in the spatial in situ platform data analysis. So it is vital to get cell typing done accurately. And once you have cell typing in place, all other applications can be built on that, like tissue atlasing, discovering function of cell types, understanding impact of neighborhood on the cell types, influence of cell interaction on disease progression, or even biomarker discovery at a single cell resolution. CosMx enables all these basic and translational research application on the top of accurate cell typing. We are launching CosMx with a broad assay menu, including multiple 1000-plex RNA panels and a 64-plex protein panel. Moreover, CosMx comes with a full customization capability. You can provide us a list of any genes up to 300 genes, and we can build a custom panel from scratch. It's like a de novo custom panel. Or, we are making a panel, it's a made-to-order custom panel for you. And on top of this, you can also customize our predefined panel. You can add up to any 50 genes to our predefined panel. So this is more on a customization of RNA. And we are currently working on a custom capability of protein. And we expect it to be ready for launch in the mid of next year, that's 2023. So if you look at the CosMx is a complete sample to answer solution with an integrated and streamlined workflow. Sample preparation is based on standard-ish protocol and compatible with both FFP and fresh frozen samples. After sample prep, you can upload sample to CosMx instrument, and CosMx instrument has an integrated high-resolution microscope for the readout. So you don't need any additional instrument. All the results or all the work can be automated on a CosMx instrument itself, and from CosMx, data get transferred directly to a cloud-based informatics portal called Atomics for storage, for analysis, as well as data sharing. So let's have a look on CosMx sample preparation protocol. CosMx sample preparation takes approximately 1.5 days with 3 hours hands-on time. However, sample prep can be automated on a Leica bond, which can further reduce hands-on time and increase consistency. It's very easy to use. There is no tissue cleaning, there is no gel embedding, there is no amplification involved. And after the sample prep, you can load and run up to 4 slides simultaneously on a CosMx system. CosMx uses in situ hybridization chemistry and automated cyclic imaging for the readout. RNA or protein, both these biomolecules targets in individual cells are identified through hybridization of targets with specific probes or antibodies, which are labeled with a unique DNA barcodes, as shown here as a readout domain. And these readout domain is decoded with cyclic imaging of fluorescently labeled reporter probes. The beauty of CosMx chemistry is CosMx needs only up to 5 targets probe per gene. And each target binding domain of these probes is very short. It's just 35 to 50 nucleotides long. And that is why CosMx works beautifully with shorter transcript and slightly degraded RNA, including FFP samples. During automated cyclic imaging, data get transferred in a real time to Atomics, a cloud-based informatics solution. Atomics provide a lot of advantages over on-prem informatics solutions. Atomics provide comprehensive visualization and data analysis pipeline, scalable compute and storage for large data outputs, easy data sharing and global collaboration. Actually, data sharing and collaboration is as easy as sending out an email. And Atomics comes with an unmatched security and high cost effectiveness. And when you look into cost effectiveness, what I mean by that is, with Atomics as a cloud-based service in place, you don't ever have to worry about establishing any IT infrastructure. So there is no upfront costs involved for buying servers or auxiliary servers or any part of it. And here is a list of CosMx data analysis pipeline, which will be available on Atomics at launch. And these data analysis pipeline include all the way from cell segmentation to cell typing to neighborhood analysis to differential gene expression data analysis. And above all, Atomics is an open system, and it empowers you to upload your own data analysis pipeline to Atomics. And thereby, it enables any custom analysis based on your own project. So you define if you want to use some other algorithm for cell segmentation, or you have any specific data analysis which you would like to do, and you have a data analysis pipeline for that, you can upload that on Atomics and use that on Atomics itself. I believe that performance of all spatial imaging technologies depend upon two fundamental attributes. First is spot calling, and second is cell segmentation. CosMx delivers highly sensitive and specific spot callings as well as accurate cell segmentation. CosMx can detect not only low expressors well above the background, but also medium expressors and high expressors, as shown over here. Its high dynamic range allows detection of hundreds to thousands of transcripts per cell. And here I have shown an example from non-small cell lung cancer FFP tissue. And in this tissue, CosMx identified an average of 300 counts per cell and maximum of 2,500 transcripts per cell. And importantly, CosMx counts show good correlation with single-cell RNA-seq dataset, as shown here on our left-hand side, as well as with the RNA-scope dataset, which is shown on our right-hand side. And these comparisons further support CosMx sensitivity as well as high specificity. So besides spot calling, cell segmentation is another important attribute of CosMx. And CosMx uses protein imaging and machine learning algorithms to deliver accurate cell segmentation. And this whole process starts with focus stacking of all these 3D images which we generate, like all different Z stacks, and we do the focus stacking of them and generate a very sharp 2D projection images. And then these images are fed into nuclear and cytoplasm and membrane based imaging modules. So what we do is, in addition to these images, we use protein-based imaging for cytoplasm as well as for cell membrane, and then we do a nucleus staining, and all these images are fed into our cell segmentation modules. Nuclear modules provide one output, cytoplasmic module provide another output, which we merge together and combine into a final cell segmentation output, as shown over here. And as a result, you get a very precise cell boundary detection. Here are few representative examples highlighting robust cell segmentation across different tissues, from your fresh frozen mouse brain to FFP, human colon, human breast, as well as human skin tissues. And we have done this for more than 15 different tissues with a very good and accurate cell segmentation. So far, I talked about CosMx RNA capability, but RNA is not the whole world. Actually, protein is equally important. In fact, both RNA and protein analysis is required to get a comprehensive tissue analysis. High-plex RNA yield detailed molecular function, but as you can see here, you need high-plex protein imaging for absolute tissue architecture analysis. And CosMx provides both high-plex RNA as well as high-plex protein analysis at a single cell down to a subcellular resolution. We are launching CosMx with 64-plex human immuno-oncology protein panel, and this human IO panel covers most relevant proteins from different biology, all the way from your B cell biology to T cell biology to checkpoint biology. And our plan is to expand protein assay menu with mouse neuroscience panel and also add customization capability by the mid of next year. CosMx protein panel goes through a vigorous antibody validation process. And these processes include pre and post conjugation individual IHC validation, followed by validation as a combined panel to check interference and the performance of the all 64 different antibodies by a trained in-house pathologist. And these antibody validation process ensures high sensitivity as well as specificity for CosMx protein panels. As of today, human IO protein panel has been tested and validated for use across multiple tissues, including lymph nodes, breast, and colon. And then a few more listed at the bottom of this slide, and we are keep testing it with more tissues to make them compatible with our CosMx protein panels. So just to summarize overall, the CosMx system delivers researchers the ability to detect more genes than any other in situ system with a large panel of 1,000 genes. CosMx has multi-omic capability with high-plex RNA, I will say highest plex RNA, and high-plex protein content, which is optimized to work across a variety of sample types, including fresh frozen and FFP. Its throughput is really scalable. You can go all the way from 2 to 20 slides per week based upon your research needs, as well as it enables whole tissue scan. You can analyze the whole tissue section in just one single run. CosMx has a scan area of 300 millimeters square. And then and on top of this, you can get a very high resolution, actually a subcellular resolution. CosMx has X and Y resolution of around 50 nanometers and Z resolution of a couple of 100 nanometers. So if you look at all these specifications, these are best-in-class product specification. But for CosMx, it is just a beginning. We are going to improve this even further as with the time. And currently, we are focusing on two major things. One is plex, and another is throughput. And we will provide more update on these parameters, which will be improved on what we already have at AGBT, which is approximately in 4 months from now. But keep in mind, there is not going to be any change on a hardware. These new capabilities in terms of increasing plex and throughput is only through the assays and chemistry part. Thanks everyone for your time, and now I will pass this on to Miranda Orr, She has a very interesting story with both GeoMx and CosMx platform. So now let's hear from Miranda Orr.
Well, thank you so much for that introduction. I'm really excited to be here and share with you some of the research from my lab, where we've been using spatial single-cell characterization of Alzheimer's disease in both mouse and human brain. I'd like to thank the funding for my lab, and in particular, the project that I'm going to be talking about today is funded by an R01 from the National Institute on Aging. My co-investigators are Dr. Dirk Keen at the University of Washington, who has provided all the postmortem human brain for this study, Dr. Habil Zair at UT Health in San Antonio, who is providing bioinformatics support. Tim Orr in my lab has run all of the GeoMx digital spatial profiling that I'm going to share, and Shiva Dakordi is a talented graduate student in the Zair lab at UT Health, who is also providing bioinformatics support. I'd also like to thank the amazing team at NanoString for helping us with the spatial molecular imaging that I'll be sharing. So over the next 45 minutes or so, I'm going to be talking about Alzheimer's disease and my lab's approach to tackling this very devastating and challenging disorder. Then we're going to shift to using GeoMx digital spatial profiling to analyze postmortem human Alzheimer's disease brain, and then talk about the next generation NanoString instrument, CosMx Spatial Molecular Imager, where we're profiling mouse models of neurodegeneration. And then at the end, share a little sneak peek of some of the CosMx data we've generated on human brain. Alzheimer's disease is the most common cause of dementia in older adults. More than 6 million Americans are living with Alzheimer's disease, and by 2050, this number is projected to rise to nearly 13 million. It's lethal. There are no disease-modifying treatments, and 1 in 3 seniors dies with Alzheimer's disease or another type of dementia. And to put that into perspective, it kills more than breast cancer and prostate cancer combined. Age is the #1 risk factor, but what research is showing is that 20 years or even more before the symptoms, brain changes associated with Alzheimer's disease may be occurring. In 2016, the National Institute on Aging and Alzheimer's Association defined Alzheimer's disease by its biology, so using the ATN framework, where A stands for amyloid beta. This is a cleavage protein from a larger protein called amyloid precursor protein, and A beta accumulates outside of the cells in plaques. T stands for tau. Tau is a microtubule-associated protein that accumulates as neurofibrillary tangles inside of neurons, and N stands for neurodegeneration. So shown here on the is an image of a brain. On the left is a healthy brain, and on the right is an advanced Alzheimer's disease brain. So you can see the dramatic brain atrophy, enlargement of ventricles, and the shrinkage of certain brain regions that are largely due to a loss of neurons. But as I pointed out, age is the #1 risk factor for developing Alzheimer's disease, and my lab is really trying to understand the connection between advanced age and Alzheimer's disease. How is it that this black box of aging can take a young, healthy brain and through the course of a lifespan, turn it into a dysfunctional tissue? For this, our team relies on the biology of aging field. And in this field of study, researchers have identified cellular and molecular processes that not only co-occur with aging, but actually drive the aging process. If they're perturbed, they can accelerate aging and drive pathology, and if they're intervened upon, they can delay aging pathologies and improve healthy lifespan. When we've identified something that we think may be relevant to Alzheimer's disease, the next step is to determine if there's evidence in people. For this, we use postmortem human brain tissue or analyze publicly available data sets. When we find evidence in people, then we can take this back to the lab and test interventions in mice. If these interventions improve cognition and brain imaging, then we move this to clinical trial. And in my lab, we are focused on tau as a driver of cellular senescence, which is a hallmark of aging that I'll talk about in this presentation. We've found evidence of senescent neurons in postmortem human brain. And when we've cleared senescent cells in transgenic mice, we've improved brain structure and function. So we've now moved to a clinical trial. So now what we're doing is using new technologies to really profile this neuropathology in Alzheimer's disease to better understand how tau is driving senescence and what cells may be best targeted by these senolytic therapies. So in Alzheimer's disease, using this ATN framework, amyloid beta is tracked by what is called thaw phase, and that just tells us where in the brain these plaques are. And tau is tracked by what is referred to as Braak stage, and similarly, this tells us where in the brain these tangles are, but it doesn't tell us anything about the molecular makeup of plaques and tangles. Now, in my lab, we're particularly interested in the tau-containing neurofibrillary tangles. That is because they more closely track with disease progression than plaques. However, not all individuals with neuropathology have dementia. That means that some people have normal cognition, but when they come to autopsy, there is evidence of plaques and tangles in their brain. And so we're asking a very simple question. Are all tangles the same? Or perhaps we can subtype them based on their molecular signature. To do this, we're using NanoString's GeoMx digital spatial profiling. And in this slide, I provided an overview of the technology. And essentially, the first step is like any other histological assay, where you stain the tissue using your morphology markers. And along with these morphology markers, you add fluorescent antibodies or RNA scope probes, depending if you're looking at protein or RNA. And these are linked with UV photocleavable oligos. You select your regions of interest or areas of interest on your tissue that you want to profile. Then, using UV light, you cleave these photocleavable oligos. So these are barcodes that then get collected and dispensed into a multi-well plate. And then they're counted either with NanoString platforms or sequenced, where you can look at proteins or up to the whole transcriptome. And because this is a non-destructive method, you can map all of these measures back to your tissue of interest. So shown here is a postmortem human brain, and we are looking at neurons in the entorhinal cortex and the CA1 of the hippocampus. And the reason is that these are regions that are prone to develop tangles early in Braak stage. Moreover, they are synaptically connected. So we can profile these neurons and see how they may be interacting or how their molecular profiles change. And for this, our morphology marker is AT8, which is an antibody that detects neurofibrillary tangles. And throughout this presentation, I'm going to be using this marker. So the magenta marks a tangle. We're also profiling neighboring neurons without tangles from the same brain region. In the particular data set that I'm about to share, we have quantified 95 proteins that span neural cell types, glial cell types, neuropathology, and protein kinase pathways. So shown here is a volcano plot where we have compared tangle-bearing neurons on the right to non-tangle-bearing neurons on the left. So anything up on the right is higher in a tangle-bearing neuron, and anything up on the left is higher in a non-tangle-bearing neuron. What I'm going to point your attention to are these top 4 proteins that are most significantly differentially expressed between these two cell types. 3 of the 4 are phosphorylated tau. Now, that may not be groundbreaking. We do know that tangles accumulate phosphorylated tau, but to us, this was a confirmation that the technology was working. Similarly, it provides evidence that in our panel, phosphorylated tau might be the most differentially expressed proteins between these two cell types. But what we're really interested in is heterogeneity in neurofibrillary tangles. So shown here is a heat map of 324 CA1 neurons with tangles from 11 cases of dementia aged 50 to 100 years old. Each column is an individual neuron, and each row is an individual protein. And so I think what you can appreciate from this heat map is there is some heterogeneity. Now I'd like to guide your attention to this purple band near the middle. This is phosphorylated tau, and you can see there's heterogeneity here as well. This is interesting to us because our prior work found a close overlap between neurons with tangles and cellular senescence, which is a fundamental cellular aging stress response. In this cartoon, I have depicted a neuron undergoing this, or a cell undergoing this complex stress response called cellular senescence. So this cell in the middle that's green is, you can think of this as a damaged cell, and it is not able to repair this damage. But it doesn't die and it doesn't continue to divide, but instead it undergoes senescence. This is a change in cell fate, where the cell actually changes its morphology. It upregulates both pro-survival and pro-apoptotic pathways, so it is stuck alive. The only way that it can be cleared is if it is cleared by the immune system. So it releases molecules that signal the immune system to clear it, but if the immune system doesn't receive this message, these pro-inflammatory cytokines and chemokines can damage the neighboring tissue, killing these cells or transforming them to become senescent. So we wondered whether or not tangles may be senescent, because we know that neurons with tangles closely track with disease progression. Neurofibrillary tangle containing neurons survive predicted up to 20 years in the brain, yet neuron loss far exceeds number of tangles by orders of magnitude. So as a first step, we used a hypothesis-driven approach where we compared neurons with tangles to non-tangle-bearing neurons in the same brain, and we published this in 2018, so I'm not going to spend too much time going over it, but we did find, if we just focused on neurons with or without tangles, neurons with tangles had a senescence phenotype. But we wanted to use an unbiased screen and more broadly look at all cell types in the brain. And at that time, the only technology available to do that was single nucleus RNA sequencing. So we looked at 140,000 cells from 76 brains with different levels of Alzheimer's disease neuropathology and used 4 embryonic brains as controls. And we created gene lists that reflect different aspects of the senescent stress response. So the initial stress response was covered by 44 genes. The stable cell cycle arrest was covered by 22 genes. And the inflammatory response, or the secretory phenotype, was covered by 48 genes. And when we asked these 140,000 cells, which of you are expressing these phenotypes, we found about 2% expressed each of these pathways. And when we asked which cells are these, we found that over 97% of these senescent cells were excitatory neurons, and they overlapped with tangles. Using our canonical stress response signature, we asked what is the gene that is contributing most to this response, and we found CDKN2D. And then for the next step, we wanted to confirm whether or not we found this in tissue. So in collaboration with Dr. Rachel Bennett from MGH, she performed RNA-scope, where in this image, the yellow are neurons and the white are CDKN2D transcript. And we found that CDKN2D was highly expressed in neurons. And then Dr. Jamie Walker, who was at UT Health in San Antonio and is now at Mount Sinai, performed P19 histology. So CDKN2D codes the protein P19. And when she stained brains, Alzheimer's disease compared to controls, she saw upregulated P19 in Alzheimer's. For our next step, we did co-fluorescence or co-staining. And so Dr. Bennett co-stained these tissues for tangles, which are shown in red, and the CDKN2D probe, which is shown in white, and saw close or strong overlap between CDKN2D and neurons with tangles. Similarly, when we did co-immunofluorescence in our lab, where we stained tangles in yellow and P19 in magenta, we saw overlap. So this was really exciting that our bioinformatics predicted a marker that we could then use and add into our profiling method. So now, in addition to using AT8 in our GeoMx profiling, we've added P19. So now we're profiling 4 different types of cells: neurons that have AT8 in magenta, with or without P19 in green, neurons with only P19 in green, and neurons that don't have P19 or AT8. And shown here is a heat map of an experiment where we profiled these 4 different types of cells. And I'd like to draw your attention to the right side of the heat map. So the green column are neurons with tangles, and the blue column are neurons with tangles that also have the senescent marker. So these would both be considered T+ with traditional Brock staging, but now we've identified a subtype that is T+ senescence. And what we see, if I draw your attention to the middle purple band, this is phosphorylated tau. So you can see there's high tau phosphorylation in both. But when we look at other proteins, there are significant difference, where the neuron with the tangle that also has a P19 marker looks a lot more like the healthy cells without the tangle than the neurons with the tangles. So our next question was, does this heterogeneity in tangles have any functional implications? And for this, I again collaborated with Dr. Jamie Walker, and we compared brains that had similar Brock stages, meaning they had similar level of tangle burden, but some individuals had cognitive impairment and some individuals had normal cognition. And what we found is in the individuals resilient to this pathology, they had lower phospho-tau at 396 and 404, and they had higher levels of synaptic protein expression, lower inflammation, and lower oxidative and energetic stress, suggesting not only is there heterogeneity in tangles, but it actually may have functional implications. We're now moving into a next generation of technology called CosMx Spatial Molecular Imaging, also developed by NanoString. So with this technology, we get subcellular resolution of these analytes. And the data that I'm about to share were done in collaboration with a fantastic team at NanoString led by Lidon Wu. For the initial spatial molecular imaging experiment, we focused on mouse brain. So shown here, I have mouse brains below the human brains, and in the same orientation where the healthy mouse brain or wild type mouse is on the left, and the tau transgenic or diseased is on the right. And you can appreciate the dramatic brain atrophy that occurs in these transgenic mice. And for this experiment, we used a 1003 plex RNA panel that spanned cell typing and mapping genes all the way through regulatory signaling. And this experiment generated amazing, fantastic data, and I'm just going to give you a little snapshot of what we found. So first, using supervised cell typing, we identified 42 different cell types and one undefined cluster. As clustering, we identified 42 cell types. And when they are mapped back to the brain, we have high confidence in these cell types and their local resolution or their location within the brain, because we know that neurons are where neurons should be in the gray matter, and we have microglia where we know microglia are. We have oligodendrocytes where we know myelin is. We then condense these cell types into more broad categories to get a broader comparison between the wild type and tau transgenic. So in this graph, we have the different cell types. The taller the bar, the more cells there are. And when we compare this wild type mouse to a tau transgenic, we can see noticeable differences that are predicted based on this model. So for example, the red bar are neurons. We see far fewer in the tau transgenic. The orange bar on the far left are astrocytes, and we do know that tau transgenic do accumulate astrocytes or have astrocytosis. So again, this gives us confidence with the method. What is really powerful about this technology is because we know differential gene expression in cell types and where all of these cell types are located, we can now identify spatial niches. So shown here are spatial niches of wild type mice. And if we compare that to the tau transgenic, you can see these dramatic differences. In fact, the tau transgenic mouse is missing 3 spatial niches. And we know what cell types are in these niches. For example, in spatial niche 2 and 5, which are significantly lower or missing in the tau transgenic, these niches are comprised largely of neurons. And again, because we know differential gene expression and where these cells are located, we now get spatial differentially expressed gene in each cell type and can make these maps. So shown here is an example of microglia. And this is a lot of data in a busy slide, and I only want to show you this as an example. So on the y-axis are individual genes, and on the x-axis are comparison between niches. So if we look at microglia and compare different niches, we can see how each niche either upregulates or downregulates that particular gene, and we can compare between genotypes. So as an example, we'll just look at ApoE. So for example, in the wild type mouse, ApoE is enriched in niche 9 versus niche 1 and 7, but is depleted in niche 1 and 7 versus niche 6. And then we compare that to what happens in tau transgenic. So in tau transgenic, ApoE is also enriched in niche 9 versus niche 1, but is depleted in niche 3 versus niche 1 and 6. And again, we have this level of data across all cell types. Another really powerful aspect of this technology is that we're able to look at multiple transcripts at cellular and subcellular resolution. So in the previous work where we were looking to identify senescent cells in single nucleus RNA sequencing, now we're essentially using that same approach, but looking at it in a tissue. So these different gene panels listed here are the same genes that we used for the single nucleus RNA sequencing. And the white boxes that I have highlighted are CA1 neurons. And if we compare wild type to tau transgenic, we can now look at these gene expression signatures within individual cells and zoom in and really ask the question, are these cells senescent? Are they expressing the gene signatures that give us confidence in identifying these cells as senescent or not? More recently, we've been working with NanoString and many of the same team members to apply this technology to formalin-fixed paraffin-embedded postmortem human brain. So something that I haven't highlighted yet, but is just incredible about this technology, is you get a morphological image of the entire tissue, and you can zoom in at subcellular resolution. So shown here in this morphology scan, histone is in blue, GFAP to measure or to mark astrocytes is in red, 18S RNA is in green, and ATA for tangles is in yellow. And then we can zoom in and look at transcripts in each of these cells. So similar to some of our proof of concept experiments using GeoMx, we've done the same thing with spatial molecular imaging in postmortem brain. So shown here are blue transcripts of immediate early genes that is in the cyan color. And these genes are transcribed in neurons when they are active. And so we're focused on dentate gyrus neurons in both healthy and Alzheimer's disease and looking for these transcripts. And in magenta are growth factors. And in green are transcription factors that would drive expression of these. And if we zoom in, what is really incredible is that if we look at the healthy tissue, the healthy dentate gyrus cells, we see immediate early gene expression, we see growth factor expression, but when we look in the Alzheimer's disease, we don't. So this again is somewhat expected. We know that the dentate gyrus cells are really important in learning and memory, and that in Alzheimer's disease, these functions are impaired. So now that we have confidence in this technology, we can begin to ask questions about gene expression that we think may be relevant to Alzheimer's disease and aging. So in this slide, I'm showing autophagy genes in cyan and lysosomal genes in magenta. We know that there is proteostasis disruptions in Alzheimer's disease. And if we compare healthy on top to Alzheimer's disease on bottom, you can see a dramatic reduction in the transcription. And if we zoom in, we have healthy now on the left and Alzheimer's disease on the right, compared to the healthy that have high transcription or high gene expression of autophagy and lysosomal genes. In Alzheimer's disease, we don't see the same patterns, except for if we look at neurons with tangles. So in this, we've now co-stained for the tangle marker in yellow, and we see that this particular neuron does have high lysosomal and autophagy gene expression, similar to the healthy controls. So again, this is suggesting that these neurons, though they are incredibly stressed with this tangle, they are surviving. So just to summarize and provide some insights into some of our future directions, we are finding that senescent cells accumulate in Alzheimer's disease, and neurons with neurofibrillary tangles are a primary source of senescent cells in Alzheimer's disease. Using GeoMx digital spatial profiling, we're now able to reveal heterogeneity across these tangles. And this heterogeneity is partially explained by senescence, and it has functional implications on dementia status. And then moving to CosMx Spatial Molecular Imaging, we're now able to use subcellular profiling to look inside these brain cell types. We're going to use this to inform on brain changes with age and neurodegeneration, both in mice and humans. So in conclusion, spatial profiling technologies are significantly advancing neuroscience research, and in particular, Alzheimer's disease research. And for translational neurobiologists like myself that requires species flexibility, we're able to profile both mouse and human brains. The technology is compatible with frozen NFFPE tissue. We're able to look at multiple analytes. I showed evidence of both multiple transcripts as well as multiplex protein expression. And what's really powerful is that we get tissue level analyses as well as subcellular resolution. So now we're able to look at this in multiple cell types, multiple brain regions, neighborhoods, and spatially resolved gene expression across tissues, cells, and niches. So with that, I would like to acknowledge my team at Wake Forest, my lab. As I mentioned earlier, Tim Orr is running all the GeoMx experiments in the lab. Again, Dr. Dirk Keen is providing all the postmortem human brain tissue, and Dr. Zaire and his student Shiva are providing bioinformatics support. Dr. Jamie Walker, I mentioned, has provided neuropathology support as well. And I'd like to thank the NanoString team for helping with the spatial molecular imaging. And I didn't mention, but Dr. Bin Zhang and Peng Zhu at Mount Sinai are providing bioinformatics support as well. Thank you so much for your time.
Thank you, Vikram and Miranda, for your time today and your important research. We would also like to thank LabRoots and our sponsor, NanoString Technologies, for underwriting today's educational webcast. Questions will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.